In this video, we're going to be talking about osmosis. Before we get started, let's get oriented to where we are. We've been talking about three core principles in anatomy and physiology. The first principle being homeostasis, the second, how molecules move down gradients, and the third, the relationship between structure and function. If we hone in a little more on flow down gradients, we've seen three forces that move molecules. Molecules will move down a hydrostatic pressure gradient, they'll move down a concentration gradient, or charged molecules will move down an electrical gradient. Osmosis is a special case of movement down a concentration gradient, and it refers specifically to the movement of water. If I eat a salty meal like a bunch of chips and forget my water, that's going to cause a disruption in homeostasis. Sodium levels in my blood are going to climb relative to the water levels. So water and sodium have to move in opposite directions to correct the imbalance. We know that hydrostatic pressure would push water and sodium molecules in the same direction, so that's not going to work. An electrical attraction could move sodium ions, but would also attract potassium ions and calcium and other important electrolytes. So the only way water will move in a different direction from other solutes is down its concentration gradient or through osmosis. In our bodies, the process of osmosis occurs through aquaporins. Aquaporins are channels in the cell membrane that allow water to pass through, but not other molecules like sodium or potassium. Most cell membranes are rich in aquaporins, so water moves freely from outside the cell to inside the cell or the other direction. In places like the kidney, the movement of water is regulated by regulating whether or not aquaporins appear in the cell membrane. Okay, let's have a look at the process. Let's take a U-shaped tube and fill it with water. We know that the water levels on each side of the tube will be the same because we have a certain amount of hydrostatic pressure from gravity pushing down on each side, and that's the same. Now, if we take the tube and separate the left from the right-hand side by a semi-permeable membrane, I've circled that in black there, that membrane allows water to pass, but not other solutes. It's the equivalent of a cell membrane with aquaporins, but not other types of transport proteins. And on the right-hand side, we'll put a glucose solution. In this case, water is the solvent. It's the substance with the greatest concentration of molecules. And glucose is a solute, a substance dissolved in the solvent. On the right-hand side of this tube, the water concentration is lower than the left-hand side. Why? Well, the glucose concentration is higher, therefore glucose molecules are taking up some of the space that would otherwise be occupied by water. Therefore, water concentration is lower. Like any other molecule, water will move down its concentration gradient from higher concentration to lower concentration, in this case from left to right. And water will continue to move down its concentration until the forces moving water from the left-hand side to the right-hand side are equal to the forces pushing water from the right-hand side to the left-hand side. Okay, osmotic pressure is pushing water from left to right, and that water has a weight. So that weight exerts a specific amount of pressure in this system. Remember that the pressure is applied in all directions, including from right to left, and the pressure is equal. So we have hydrostatic pressure moving all of these molecules from right to left, but only water can get through the membrane. Osmotic pressure is pushing water from left to right. When those two forces are equal, there will be no net movement of water. Okay, let's talk about the terms osmolarity and tonicity. Osmolarity refers to the amount of osmotic pressure a solution would exert. a solution with a greater concentration of solutes will exert more osmotic pressure than one with fewer. So osmolarity refers specifically to the number of particles dissolved in a solution. This blue solution we're looking at here has some red dots in it. Those are solutes like glucose we saw before. If we increase the number of solutes, doesn't matter what type of molecules they are, it will increase the osmotic pressure. So if we had sodium, potassium, chloride, different proteins, it doesn't matter. Each of those will increase the osmolarity of a solution. Now, what happens if we take a red blood cell and dump it into this solution? Tonicity refers to the effect the solution would have on the red blood cell. If we dump the red blood cell into an isotonic solution, that means the amount of water entering the cell will equal the amount of water leaving the cell. So if we imagine that no solutes in our solution cross the cell membrane and nothing from inside the red blood cell goes back out into the solution, then the osmolarity of the solution we dump the cell into would be the same as the osmolarity inside the red blood cell. Let's keep that same assumption and look at a hypertonic solution. If we took a solution with high osmolarity, none of those solutes entered the cell, then more water would travel out of the cell than in, and the cells would become crenated. They would become dehydrated. And the opposite is also true. A hypotonic solution has few solutes, and so water would rush into the cell. On the face of it, it might seem that tonicity and osmolarity are the same thing, but that's not necessarily true. Let's imagine we're going to dump the red blood cell into a solution that's isoosmolar. It has only these red molecules, and the concentration of those red molecules in the solution is equal to the concentration of solutes in the red blood cell. But these red molecules can freely cross the cell membrane, and nothing happens to those molecules when they enter the cell. So in this case, as soon as we dump the red blood cell into solution, these red molecules are going to cross the cell membrane down their concentration gradient until the concentration of red molecules inside the cytoplasm is equal to the concentration out. What this means is the solute concentration inside the red blood cell increases. As that happens, the osmotic pressure exerted by the cytoplasm increases and water rushes into the cell. Eventually, the cell will burst. Okay, to summarize, osmosis is the diffusion of water down its concentration gradient from high concentration to low concentration. Solutions with greater solute concentration have greater osmotic pressure. Tonicity refers to the effect a solution will have on a cell, and that's often what we care about in medicine, because if we administer a solution, the tonicity is going to define the effect it has on intracellular versus extracellular fluid volume.